by scientist Gregor Mendel. And he performed some of the experiments on the basis for studying the hereditary. Now, remember, in our study, we are going to study about only two types of experiments. Experiment with single character, experiment with the double character. In fact, in the case of heredity, not only single or double character are there, large number of characters are included in the field in heredity. But over here, for 10th standard level, we shall understand only about the single character heredity and obviously the double character heredity. Now Mendel selected the garden pea plant. The scientific name is Pisum sativum. That is, he took that as an experimental material because so many generations have to be studied in order to understand the process of heredity. The pea plant shows various characters, at least two contrasting expressions of each of the characters. That is why he selected especially the garden pea plant. Now, first of all, let us understand about the Mendel's experiment of the single character. First of all, he took two different types of plants. One of them having a tall pea plant having capital T traits of DNA. Whereas the other plant was of a dwarf pea plant and that was small letter TT traits of the DNA. Now, here the characteristics that he studied was the tallness and the dwarfness. All the plants of the F1 generation were found to be tall. That means when he crossbred a tall plant, tall parent with a dwarf parent, then whatever the offsprings were obtained, those offsprings, those new generations, that is F1 generation, were all of them tall. There was not a single dwarf or even a medium height that was produced in F1 generation. Wonderful, isn't it? That means only one parental characteristic is expressed in the offsprings. That is tallness in this case. So F1 generation of the plant, when they are self-fertilized, that means a particular plant of F1 generation crossbred with another particular plant of the F1 generation. That means F1 cross F1. Then we get F2 generation plants. He found out that 75% of the plants were tall and 25% of the plants were dwarf in this F2 generation. So from F1 generation, both the genes, that is of the tallness and of the dwarfness, that were inherited. Let us try to understand this in the form of a figure. So the parental plant, tall plant, having capital TT characteristics of the gene, crossbred with a dwarf plant with a small tt characteristics of the gene. That gave rise to the F1 generation. And that F1 generation, we can understand that mathematically also. But let us first of all identify that what type of plants were obtained. You know that all the plants that were obtained were tall plants. Now, here you can see that how did we get this particular combination. For example, if we combine this T with this T, what are that we are going to get? We are going to get it as TT. Same way, the same T is to be combined with another T. And that is also going to give us TT. And same in the same another manner, T with T, that is going to give rise to TT, capital and small letter. And another capital and small letter T, that is going to give rise to another same thing, that is TT. But let me tell you, all the plants, all of them were what type of plants? They were all tall plants. That means capital T must be the dominant character. Now, F1 is crossbred with the same F1 generation plant. And in the same manner, what we are going to get is F2 generation. And that is a tall plant, a tall plant, a tall plant, but a dwarf plant. How did we get that tall plant? Because if you combine F1 generation T with capital T, and that is going to give rise to. Now he crossbred two different plants of F1 generation, that is F1 cross F1. That means a tall plant of F1 generation is crossbred with tall plant of F1 generation. And what do we get? We get F2 generation. And in F2 generations, you know that 75% of the F2 generation is tall plant, whereas 25% of the generation is a dwarf plant. How did we get this? Let us try to understand. That when capital T, a DNA characteristics of the tallness, is crossbred with another capital T, then we obviously obtain TT, obviously a tall plant. Same way capital T is to be crossbred with small t of the other plant, and we get capital T and small t, again a tall plant, because capital T, as I told you, must be the characteristic of dominant characteristics of inheritance. Same way, now T with capital T, and that is a tall plant again. But a small t crossbred with a small t, and that is going to give rise to a pair of small t generation, small t DNA, TT DNA. That means a dwarf plant. So, I hope you understood that the F1 generation of single characteristic experiment, all of them were tall plants. But in F2 generation, 75% of them were tall plants, whereas the 25% of that was a dwarf plant. Now, from this, it indicates that capital TT or capital T and small t, that genetic constitution is for the tall plants. Whereas for the dwarf plant, both the traits, that is small t and small t, that is the constitution of obviously the dwarf plant. Only one gene, capital T, is sufficient for expression of the tallness. Whereas both the T's should be small letters, and then only we can say that the characteristic would be, that characteristic inherited would be dwarfness of the plant. That means, as far as the characteristics of only tallness or dwarfness is concerned, then capital T is the dominant gene, whereas the small t is recessive. Because even if small t is present along with capital T, what do we obtain? Obviously, a tall plant. But if it has to be a dwarf plant, compulsorily, both the things should be small t, then we get a dwarf plant. Now let us understand Mendel's experiment of two characters. Now Mendel selected what? You know that pea plant. But over here in the pea plant, he selected two different characteristics. Yellow and round seeded plant as one of the parent plant. And he crossbred that with green and wrinkled seeded plant, that is YYRR, as another parent plant. Capital Y stands for the yellow color. Small y stands for green color. Round, that is represented by capital R, and the wrinkled is represented by small r. And obviously they will be found in pairs, and that is why YYRR in capital for yellow round and small letters for green and wrinkled seeded plants. Here the dominant gene for color is Y. That is Y of yellow color is the dominant gene. Whereas recessive gene is small y, that is for the green color of the seed. Whereas the dominant gene capital R is for the round shape, and obviously the recessive gene, that is small r, is for the wrinkled shape of the seed. Now all these things, all these characters are present in the parent generation. That means these are the characteristics found in one of the parents, yellow and rounded seed, another parent, green and wrinkled seed. So YYRR, when crossbred with YYRR small letters, that is going to give rise to a combination of capital Y with small y and a capital R with small r. Whatever the number of combinations you are going to make, you are going to get the same answer. That means YYRR with capital small in order, in a chronological order, in the same manner. So the F1 generation would be obviously capital Y, Y, small letter, and capital R and small r. That would be the characteristics of the F1 generation. So in all the F1 generations, what type of seeds do we obtain? Capital Y is present, dominant gene, and that means it must be yellow in color. And capital R, the dominant gene, that means it must be round in shape. So all the plants of the F1 generations, they were having yellow and round seeds. So let us try to understand the same thing. When a yellow round seeded plant is crossbred with green wrinkled seeded plant, then what do we obtain in F1 generation? We always obtain yellow round seeded plant. So that means what we are going to get is yellow round seeded plant in case of F1 generation. But now, if the seeds color of yellow and green seeds of different shapes, that is of round and wrinkle, they are inherited independently in this case. Remember, yellow and green do not mix each other. They are inherited separately. Exactly the same way the round shape and the wrinkled shape they are also inherited separately from each other. So the plants of F1 generations were then self-pollinated. That means the self-pollination of any of the plant of F1 generation was carried out. What happens? Two new combinations were observed, which were not there in original parent plant also, or in F1 generation also. Yellow and wrinkled seed, a completely new character. And along with that, the green and the round seeded plants, that was observed in the F2 generation. So this came into an existence by self-pollination of F1 generation plants. So that is how the F2 generation was obtained. So F1 cross to F1, that gives rise to F2 generation. And let us try to understand that when F1 generation is crossbred with another plant or self-pollination of F1 generation, then what do we obtain? F2 generation. And that F2 generation will have different combinations. Yellow round seed or else yellow wrinkled seed. Green round seed and green wrinkled seed. Now, what type of plants do we obtain? If we try to substitute these values mathematically also, then first pair when crossbred, that is going to give rise to yellow round seeded plant. Second pair when crossbred, that is also going to give rise to yellow round seeded plant. Third pair, yellow round with green round, that is going to give rise to yellow round. And in the same way, the fourth pair, that is also going to give rise to yellow round seed. See, yellow round seed, that is a dominant seed as far as this part is concerned. But now, when we consider yellow wrinkled seed with yellow round seed, then that is going to give rise to yellow round seeds once again. Yellow wrinkled with yellow wrinkled, and that is going to give rise to yellow wrinkled. Yellow wrinkled with green round, and over here that is going to give rise to yellow round seeded plant. And same way, all the different combinations that we obtain, that is going to give rise to two different new characteristics. And those two different new characteristics are what characteristics? The original plant never had yellow wrinkled seeded plant. The original plant never had green rounded plants, the seeded plants. And then also we obtain in the F2 generations those types of plants. So the new characteristics are obtained. What are those new characteristics obtained over here? That is yellow wrinkled seeded plant or else green round seeded plants. Now if you observe in this particular table, what we are getting is, we are getting 9 yellow round, 3 yellow wrinkled, 3 green round and 1 green wrinkled. If we consider the first ratio, that means 9 yellow round with 3 yellow wrinkled. That is one original characteristic, another new characteristic. Then the ratio is 9:3, that is 3:1. And exactly same way, green round, that is the new characteristics, and 1 green wrinkle, that is the original characteristics. Once again the ratio remains same, that is 3:1. So the new characteristics are obtained in the form of 3:1.